This protocol can be applied to any process that results in double-stranded DNA breaks. It yields detailed, quantitative data on kinetics. Although this method has single-molecule resolution, it measures up to 1,000 DNAs in a single experiment, thus providing good statistics. The primary application of this method is to study the mechanisms involved in DNA binding and modification. For example, we are interested in studying DNA target search, which is relevant to all DNA binding proteins.
When attempting this technique for the first time, remember that single-molecule tethers are delicate and once formed must be handled with care. Proper preparation of functionalized surfaces is essential. There are a few tricks for making the flow cell, as well as switching sample tubes during data collection. Visual demonstration can help someone who is new to this procedure.
To wash the cover slips, place them in staining jars and sonicate them with ethanol for 30 minutes. Thoroughly rinse them with deionized and distilled water, then immerse them in 1 molar potassium hydroxide and sonicate them for 30 more minutes. Repeat the wash sequence, making sure to rinse the cover slips with water after each wash. Store the cleaned cover slips in staining jars with water. Use a clean razor to cut 8 cm long loading and exit tubes and insert them into holes in a clean glass slide. Epoxy the tubing and let cure for 5 minutes to secure it. Apply double-sided tape pre-cut with the channel pattern to the glass slides and smooth it out with plastic forceps to achieve a good seal. Peel the backing off the tape and apply a clean cover slip